Good afternoon, everyone. Welcome back to yet another JMitra Scientific webinar on the subject of rethinking sepsis diagnosis, the role of automation. Now, sepsis continues to be a leading cause of mortality in critical care settings, where timely diagnosis can mean the difference between life and death. As diagnostic laboratories increasingly adopt automated platforms, the ability to rapidly and accurately measure sepsis biomarkers has become central to early detection, risk stratification, and effective clinical management. Well, today's webinar, The Role of Automation in Sepsis, explores how automation is reshaping this critical diagnostic landscape. We are honored to welcome back Dr. Upasana Bhumbla. Bhumbla, professor in HOD microbiology and lab director at Adesh Institute of Medical Sciences and Research, Bhatinda, as she says, her expertise on this vital subject, right? Less from me, more from you, ma'am. Over to you.
Hello, good afternoon. Thank you so much, sir. Thank you, first of all, JMitra, for giving me this platform for talking on this sepsis diagnosis. Needing it to be the need for an hour, and we all have been talking since our post-graduation days, our graduation days, so how things work and how things should be working. So I'll take you to a brief, a rough about, a thought about what I have inculcated in over these years of my 10 years of experience. So thank you one and all. Thank you, JMitra. And I'm Dr. Upasana Bhumbla. I'm professor and head, and I'm the lab in charge, lab director at the Central Laboratory at the Institute of Medical Sciences and Research, Bhatinda. So if I have to share, I hope the screen is visible to everybody. So just 1 second. So sepsis, as we define, it's the organ dysfunction caused by the body's extreme overactive immune response to infection. So definitely it's very difficult to recognize and to diagnose at a very early stage, because the clinical picture, how it comes to us, patient is having fever with chills, he's disoriented, there can be shortness of breath, feeling of severely sick, confusion, tachycardia, patient with extremely ill condition, pale skin. So multiple signs and symptoms which definitely present to us as a septic parameter. And very interestingly, many times it is confused with other conditions. So by the time the patient with this rapid breathing or with low BP, with tachycardia, with fever, so before diagnosing or labeling the patient to sepsis, we have multiple other things to thought about, and yet the specific symptoms sometimes are missed, sometimes the diagnosis is missed. So how do we talk about? So the need for the early broad spectrum antibiotics in sepsis may increase antimicrobial resistance. So there are people who believe in short-term therapy and earlier outcomes, and there are people who definitely belong to our category who wants to have a good therapy and with less number of antibiotic resistance. Because yes, what are we giving to our future generations? Do we have a lineup of reserve drugs? Do we have a lineup of restricted antibiotics? And what better outcomes we are giving to the society? So empirically, everybody starts with the treatment, but have you ever thought that this particular antibiotic is actually fulfilling the supplement for an antibiogram or for a microbial resistance to prevail? So please adjust antibiotics to narrow spectrum as early as possible and wait for your laboratory results till the time it is diagnosed and can be treated accordingly as per the organism wise. Of course, the burden in India, for how many days, the length of stay matters, how costly the healthcare system is. You talk abroad, of course, it talks to you in dollars, in India, in INRs. Nowadays, the threat of the intensive care units fees, the length of stay in ICUs, it actually broadens me to think that how better we are preserving or conservatively we are managing the patient, and at the same point of time reducing the pocket constraints so that we are giving the best healthcare provisions to our Indian scenario. So sepsis complications and hospital readmissions, that is one directly proportional to increased cost to the healthcare systems. So how important is our blood cultures? So when we talk about blood cultures, we just say that send in a blood culture, okay, to a microbiologist. Maybe, she'll report by the time I'll start with my empirical treatment. I think we have answers to this. We talk only about a single pair. We talk about a single blood culture. We talk about just one random blood culture set will try to suffice a point. But the number of blood culture sets improves the yield of pathogen recovery, minimum of minimum, with 2 blood culture sets, the increased number of pathogen, and how easily we can diagnose, and we have a set of investigations what we line up. Beyond that, we can investigate a person. So a single blood culture bottle or set should never be drawn from adult patients, since the practice will result in an inadequate volume of blood culture and a substantial number of bacteriemias can be missed, which is, I'm sorry, a very sorry figure for our healthcare system. So the number of blood cultures and the volume, why does it matter? Single blood culture will miss 10 to 40% of your bacteremia patients. There will be inability to distinguish blood culture contamination from the true bacteremia, which is very, very common. And almost 40% of blood cultures drawn in hospital are generally single. We are just saying, sending in a single patient is not ready to get it pricked, or we don't have the resources with us, we don't have the blood culture bottles. So all these constraints, when you're putting up a single blood culture bottle to us, we don't suffice with it. So what are the current recommendations? Please at least send us 2 sets of blood cultures, typically consisting of 10 ml of blood in an aerobic and an anaerobic bottle. I'll be talking further what a set includes to be. 40 to 85% of blood cultures contains inadequate blood volume because the nursing staff, the healthcare staff which is providing, so they are just pushing it up, and you have an idea saying that, okay, that 1:10 ratio is never ever followed. So how much ever the blood you are pushing up, that 100 ml of fluid, the typical soya broth, which is generally there in the automated blood culture bottles, is it actually sufficing? So is it actually allowing the bacteria to grow, whether it's an aerobe or an anaerobe? And why do we require repeat blood cultures? So without signs of infection or inappropriate or inappropriately taken blood cultures, it won't be of much help to our system. How much volume is required? To talk about, we need to have first and the foremost, when we say a paired blood culture, preferably have a aerobic and one anaerobic bottle, each bottle comprising of 10 ml of blood at least, so that the minimum quantity, that is a one colony forming unit of blood per ml of blood, which can be picked up, and one colony also will be probably giving the answer whether the patient is dealing with bacteremia or septicemia or not. Ideal can be 2 to 3 sets, but of course, if the patient, or sometimes in pediatrics, it is very, very common, it is very difficult to take 2 sets of blood culture. So a minimum blood culture, 1 set, including an aerobic and anaerobic bottle, can suffice your point. But the best one or unique one can be 2 to 3 sets of blood cultures, which comprises of 2 aerobic and 2 anaerobic bottles, will be of much more importance to us. As I said that in pediatric, according to patient weight, how much is the blood volume required? So pediatric, otherwise also we have a constraint saying that we are not able to get patients' blood sample. It's not easy to get blood extracted from a pediatric patient. So think wisely why it is important, what is the use, how it will be helpful to us to label the patient to be in septicemia or not. What is an ideal blood culture set? A paired one, which comprises of one anaerobic and one aerobic. So there are number of anaerobic bacteremias generally we miss in our day-to-day practices. Generally we miss. So here are few of the studies what we have gone through. So there's a study from JCM, from American Society of Microbiology, which in blood culture sets, we were talking about the multiple microorganisms, including the aerobic and the anaerobic, how they could have helped us out. There was a study done in November 22 to 23, which evaluated that Staphylococcus aureus from anaerobic blood cultures was also demonstrating total value, leaving it a p-value of 0.009. That was a faster mean to positivity, time to positivity. Easily we could extract out Staph aureus. Interestingly, we had from anaerobic bottles, we isolated Staph aureus and Proteus mirabilis also, and only aerobic bottles, though we label it saying that only the facultative aerobes can be reported, like Staph aureus or E. coli, but much importantly, many of the anaerobic bottles also could help us growing some aerobic bacteria. So mainly we are saying that we are sending a set of one aerobic and one anaerobic blood culture bottle. There are places where paired blood cultures are definitely sent. A set of blood culture is definitely sent, but sometimes it is just comprising of only and only aerobic bottles and not anaerobic bottles. So sometimes only the aerobic bacteria you're just trying to label it out. Why not anaerobes? And there is a mystery saying that there are many anaerobic bottles which have picked up the aerobic organisms also, giving us a varied picture. These are some of the studies which highlighted that 18 facultative anaerobes and 2 strict aerobes were definitely grown, starting from Staph aureus, from Coagulase negative Staphylococcus, from Enterococcus, Faecalis and Faecium. We had Pseudomonas, non-lactose fermenters like Acinetobacter, many other enterobacterials like Klebsiella pneumoniae, Enterobacter cloacae, Escherichia coli. So multiple organisms could be highlighted when we have a paired blood culture. So commonly recovered to our instance, we realized Pseudomonas being a purely aerobic organism, we could isolate it from an anaerobic blood culture bottle also. And also many of the Candida species were also able to be identified from this blood culture, anaerobic blood culture bottles. So you can't miss up saying that only aerobic bottles can have, we have the generally GNBs and GPCs with us, and we can miss out. There can be the same aerobic and facultative anaerobes can grow up in an anaerobic bottle, and giving to your instance what you have missed in aerobic culture bottle can be picked up in an anaerobic culture bottle. So what are the guidelines? The typical guidelines when I talk from, if we talk about American Society of Microbiologists or we follow the European guidelines, typical we are saying that two paired blood cultures, including one aerobic and one anaerobic bottle, will increase the frequency of having the more number of isolates, the more number of uncommon bacteria also can be picked up during these if you're ruling out a sepsis patient. Especially in the last one from the New South Wales, it is saying one aerobic and one anaerobic bottles, or two aerobic and two anaerobic bottles, basically 4 bottles and 40 ml of blood drawing from a patient, which is difficult from a pediatric, but from the adults, at least you can line up and you can have a proper diagnosis for a patient. Right? These are some ASM, European guidelines, New South Wales and UK standards. We talk about the blood culture systems. So what are the advantages of anaerobic culture bottles? Double volume of blood that is cultured. You can recover anaerobes and plenty of anaerobes, from gram-positive anaerobes to gram-negative anaerobes that can be picked up in blood culture bottles. Improved detection of facultative anaerobes like Streptococcus and Enterobacterials. Faster detection in anaerobic conditions, and only in specific anaerobic bottles. Maximum time in the previous study, what I was showing, which was done in 173 isolates, 16% of streptococci and 17% of enterobacterials could be picked up. Although aerobe, a recent study showed recovery of Candida glabrata also, which is faster in anaerobic. As I stated, Pseudomonas was isolated, Candida was isolated, Candida glabrata, to be specific, was isolated from an anaerobic blood culture bottle. So what is the diverse pathogenicity? To label it, of course, two paired blood culture bottles, one aerobic, one anaerobic, the maximum significant value, or else two aerobic, what we are trying, but yes, we will be missing up. But the minimum point, what we say, if we label out one aerobic and one anaerobic, two aerobic bottles, the study what we, from the JCM study, what we correlated, there were 100 and 1186 pathogens could be actually isolated in cross when we spoke about taking up both the parameters on one side. So blood culture for fungal isolation, yes, that is also very important. It plays an important role. There's a Isham study which was done in 2022, and contribution of anaerobic blood culture for the revealing of Candida glabrata. It was a multicentric study wherein they said that septicemia ruled by Candida glabrata could have been very helpful for healthcare setup to go for the basic investigations, and you line up rather than thinking only on the bacterial point, you could actually treat the fungal infection also. There was another study by ASM. There was efficient recovery of Candida auris and 5 other medically important Candida species from blood cultures containing clinically relevant concentrations of antifungal agents. Though we have the MIC values of the antifungals, also the 5 basic antifungals, patient was well treated, and this multicentric study, which was done in Italy, was one of the turning points, saying that why not, especially on fungals, we should not miss them. And Candida auris being one of the notorious candidas should also be picked up on a very routine basis. Bringing coherence to MICs in clinical practice. So when we talk about in vitro and in vivo, so we talk about antibiotics, how we challenge, how we process them, how we are doing the AST. That is the methodology what we follow in lab to test microorganism in the presence of various battery of antibiotics what we put up with their MIC values, and also some with the distribution also. And in vivo, actually, when we talk about this PK/PD thing, so one word, what is PK is pharmacokinetics, dosage concentration in serum, what the body does to the drug. And what is PD? Pharmacodynamics, concentration at the site of infection, biological effect, what it is. So what exactly the drug does to the body. So there is panel of things what we talk when we talk about AMSP. Antibiotic misuse and overuse, how antibiotics are actually tapered, what is escalation therapy, what is de-escalation therapy, what is antibiotic therapy, what are the modern tools for diagnosis of this ASTs, what are the MIC values, what is diffusion. So various breakpoints when we talk about. Very quickly, if at all, I can take you to the pathways. So we have the initial antibiotic therapy, empirical treatment, what generally clinicians do. So what journal pattern, what is a local antibiogram it is talking about? What is your hospital antibiogram, your hospital flora is actually talking about? And do we have any prognostic, any rapid tests? The basic parameters, there are, before labeling it, there are initially within 90 minutes, you can have a biofire rather than waiting for a Vitek report for having the MIC values. You can have biofire, the rapid testing methods. Then we come to the targeted therapy, waiting for the lab results, waiting for the microbiological results between 36 to 48 hours. So what lab is giving us, what bacteria, what we are diagnosing. Then there is something called as personalized therapy, MICs with TDMs, that is therapeutic drug monitoring, how we are escalating or de-escalating, how we are, as per the MICs, I'll be taking up later what is MIC and how do we interpret it, and what is the monitoring therapy what we are following. So are we looking for any other different sepsis biomarkers? We look for bioprocal, we look for any LBMs. So what are the different biomarkers? So this is how the strategy should have been following. So while minimizing the adverse drug events, look empirically what has started. Was it actually suffice when the actual culture report has come? And when the culture report has come, what are the MIC values? What is the therapeutic drug monitoring? What a clinician or a microbiologist would have helped to a patient? And any other sepsis biomarkers we are ruling out. So this is one enormously used gadget, Vitek 2. We definitely get the fastest routine AST isolates as soon as possible. We have the organism names, we have the different MIC concentrations, we have the identification, we have the different biochemicals according to which the organism is identified. Most efficient workflow in the market, I said from north to south, we have this machine updated, fully integrated ID and AST solution for our problems. So when we talk about MICs, what is MIC? It is the lowest antimicrobial concentration that inhibits the growth of bacteria or a fungus. And definitely it's a measure of susceptibility of a pathogen to an antimicrobial. So the aim of MIC is to predict the likely treatment success or failure to a chosen therapy. In my words, if it's all I put it, minimum inhibitory concentration, MIC, is a phenotypic endpoint in a defined and a standardized way, but with a high relative system. So when we talk about MICs, we definitely have this our notion saying that, okay, higher the MIC, lesser the antibiotic will be working. Lesser the MIC, higher the antibiotic might be working. So let's talk about how it works. So we have different parameters where agar diffusion are inappropriate, where actually this can be helpful, where actually it cannot be. So when we talk about staph, there are multiple drugs which cannot be done only by diffusion, like vancomycin, daptomycin, telvinafocin. We can't have diffusion and just say that this is a sensitive drug. These breakpoints are very helpful for the patient. Then for the beta lactams, especially for BLNRs, for Streptococcus pneumoniae. Generally, Streptococcus are very, very sensitive to penicillin. So how well we are diagnosing them? Detection of low level of antibiotic resistance, especially in Salmonella. We just say that fluoroquinolones are very much resistant. We have multi-drug resistance. We have drug resistance to fluoroquinolones. Is it the drug of choice? MIC creeping, which is very common in Staph aureus. And if you talk about how we are preserving this broad spectrum antibiotics, especially in enterobacterials. So when we talk about all these, we are talking about, importantly, Salmonella. Are we actually diagnosing the fluoroquinolone drug resistance in Salmonella typhi? How well the testing and reporting of Salmonella is done? Does the routine susceptibility testing is indicated in Salmonella species? So what is the drug of choice? If fluoroquinolones, if ciprofloxacin or levofloxacin, what is the MIC values? Pifloxacin, the surrogate marker, whether it was diagnosed or not. So basically, no single test detects resistance resulting from possible fluoroquinolone resistance, especially when we talk about the Salmonella typhi. Otherwise, yes, Salmonella typhi, septicemia, you try to label that second, third generation cephalosporin ceftriaxone is a drug of choice. So does MIC predict clinical outcomes? Yes, of course. For beta-lactam and BL inhibitors, old and new lower MICs seems to be important to treat serious infections. There's one word about used to breakpoint to MICs, BMQs when we talk about. So breakpoints are concentration that separates different populations of bacteria. And to rule out MICs, broad dilution is the most important goal, what we talk about. Very briefly, if at all, because CLSI is a world in itself. UCAS is a world in itself. So clinicians, when they talk to us saying that MIC values, what is this SDD dose, so there's a typical picture. This is one table 2A for enterobacterials from CLSI been taken up. Here the value it is showing less than 2 microgram per ml for cefepime, and it is saying that 1 gram, which can be CLSI very beautifully it writes for how long, what can be the dosage, and for how long you have to give. So 1 gram administered every 12 hourly. So then there is something called as susceptible dose dependency, right? The SDD. So when you have cefepime SDD of 4 micrograms, so how do we divide the dose? So 1 gram administered every 8 hourly and 2 gram administered every 2 hourly. Of course, there can be a value saying that as MIC of 8, which is quite higher. So how do we here the PKPD values, what body has done to the drug and what drug will do to the body. So 8 microgram, how will you divide it? 8 gram administered every 8 hourly. How you are giving it? It is completely from a clinician point of view. You need to identify what is the MIC value, what are the BMQs. Sometimes MIC is 2, whereas the susceptibility if I remember of ceftriaxone in a gram negative bacteria, it was showing it is 2, and the susceptible value was almost 8. So you were saying BMQ of 4, 8 divided by 2, 4. So it was talking very nicely that BMQ of 4 will be very helpful for a patient to go for the better antibiotic like ceftriaxone. This is something from UCAS. UCAS doesn't follow the thing of SDD, susceptible. Here we call it as susceptible intermediate, or the UCAS exposure um, uh, in routine with SIE in UCAS. It is of, so the values, they are diagnosed as standard dose and higher dose. There they're talking about either a MIC or a susceptible drug dosing in CLSI. Here, for the PKPD values we talk about in UCAS, we are talking about SIE. So there are some organisms where you can't have disk diffusion methodologies. Only the MIC values has to be diagnosed rather than going to a disk diffusion. There are many times, I think every day in a microbiology lab or some smaller setups or a standalone lab, sometimes we have tested vancomycin with a disk diffusion. Or for streptococcus, you are having this for amoxicillin, for amoxiclav, for cefotaxin, and for the third generation cephalosporins. You are saying you are doing a disk diffusion, and that will probably justify writing just S and R. No, you need to have the MIC values, then only the drug can be started. Because these are high-end drugs, these are restricted drugs. You just can't start in a drop of hat and say that this can be helpful. So what does CLSI recommend? So MIC values determined may be reported directly to clinicians for the patient care. However, it is essential that an interpretive category of sensitive, intermediate, or resistant also be provided routinely to facilitate understanding of the MIC report by the clinicians. Zone diameters should not be reported because it doesn't give any interpretation to the categorization. Recommended because clinicians won't be aware of it. Even some of the microbiologists, it is actually the parameter. It should not be reported. Recommended interpretive categories for various and the zone breakpoints are based on evaluation of data, which is clearly demarcated well in CLSI, which is read and I think dealt with every microbiology lab in India. So the reference gold standards remains CLSI, and for the breakpoints, we have the broth micro dilution for the MIC values. Macro dilution, that is definitely, it is more cumbersome. E-test, log values from zero point 0.5 to 1.5, but though it is expensive, it's not routinely so very projected well. So for Vitec, MIC values with good MIC values, which follows the criteria of CLSI, we should have one of these parameters in our hands. So we have accurate diagnosis with the labeled MIC programmed Vitec 2. It can give the organism identification. It can give you reliable ASTs with good, might be coming up with this breakpoint to MICs. BMQ values also will be, or from the same values, you can calculate the BMQs at your level in making it one Excel sheet. What are intrinsically resistant? We can have this SDD dosist. And there is some take home message which I really want to share it out here. What are the antibiotics which should not be reported in ASD? Very commonly we see tigecycline being reported in blood. No, no, no. It's a severe no, no. In CSF, very commonly first and second generation cephalosporin, colistin, oral agents, doripenem, carbapenem, this cannot be given as per CLSI. Many abscesses, we are labelizing aminoglycosides. It's rational use. It should not be. For lungs, for lung abscesses, daptomycin should not be reported. For children, again, fluoroquinolones, tetracyclines, chloramphenicol, this should not be used for. Urine, very commonly tigecycline, chloramphenicol, clindamycin. And I think fosfomycin nowadays, it has become a fashion. Fosfomycin is only the drug of choice in Escherichia coli and Enterococcus. Otherwise, it's not the drug of choice for all the enterobacterials and all the non-lactose fermenters. In pregnancy, avoid tetracyclines and reporting of fluoroquinolones. So it's a take-home message. And which antibiotic to be reported for the specific sites? If we are suspecting to have some SSI or soft tissue infection, please report tigecycline there rather than reporting it in blood or CSF or anywhere else. Please report it in SSIs. Then nitrofurantoin in lower UTI, especially by, as I said, E. coli and Enterococcus faecium. In Enterococcus faecalis. Nitrofurantoin in lower UTIs, it's one of the oldest and the good drug which is being used, and beta-lactones is not for lower UTIs at all. It is not to be reported. And amikacin not to be reported in Pseudomonas except in urine sample. So please make note of these things. These are very much readily available in CLSI. It has been reported by one of our senior faculty, how he has jotted down what is supposed to be reported and what is not to be supposed to be reported. So if I say that what can be your take home message, first and foremost, when we talk about septicemia, we need to have the paired blood culture. We need to have paired with one aerobic and one anaerobic. Then we need to find out what are the MIC values, whether you're following CLSI, whether you're following UCAS, and we can have the BMQ values and what exactly the organism identification and AST monitoring, escalation therapy, de-escalation therapy, and then targeted monitoring. Lastly, the use of biomarkers can be very, very helpful for ruling out different set of organisms causing septicemia. With that, in a very short thing for my presentation, this was all about our septicemias, how automation using Vitec, how automation using MIC values, how
Sorry, ma'am, we seem to have lost your voice.
you hear me? I think it is fine. Hello?
We can hear you loud and clear, ma'am.
I hope the presentation, during presentation, it was
Oh,
there.
It was absolutely clear. And
Okay.
thank you so much for the the detailed approach, because everybody knows a lot of things, but putting it together takes a lot of effort and conviction, if I may use the right word, conviction. And your conviction is evidently quite visible in in this line of thought process around sepsis, around the use of automation. I had a question because the Whitec or the loss, there is a cost differential, ma'am.
Okay.
It's a cost, and it's about 2.5 times or 3x, I think, the cost differential. So what is in the interest, because there is a cost that is if we go in the direction of automation, uh, are we having a are we shifting the cost from the patient outcomes to the diagnostic product?
My fourth slide spoke about the length of stay. Definitely. So distribution, definitely it's an older method. We are never ever saying don't go for the conventional ones. It is definitely there. My second point saying that please wait for the microbiological culture reports, because microbiologists play a very important role in giving up what organism can be there. So if we have the right organism, right antibiotic in your hands, why this length of stay in a hospital will be varying? So from there, the cost will be reduced. So
Okay,
if you are actually, I'm talking about setups who can have this bare minimum with the MIC values.
right.
Why not to go with this minimum standard? Why not?
And okay, a lot of pathogens are also viruses, atypical organisms, fungi. Some of them aren't culturable at all, right? And conventional
There
culture,
have been saying that yes, when we talk about anaerobes, 100 times it is missed. That's the reason I'm saying when we are talking about paired blood culture now with this news, people have developed AI. We are asking computers to work for us. So if you're asking so much of, as you say, that informed or well high tech technology in your life systems, why not to deal with microorganisms? Anaerobes are 90% of the time it is not touched. I don't know how many microbiological labs in India itself will be only talking about a pair talking about one aerobic and one anaerobic. I don't think so. It is followed at maximum areas. I talk about Punjab. I don't think so. At many places, it is being followed. But if we do it, I think many answers will be corrected, which are sometimes misread or mistreated.
before we started this webinar, ma'am, we talked about someone has to come out with that idea. Somebody has to put that thought on the table, because
Yeah,
today's radical idea becomes tomorrow's common. Like, hey, I always knew this, because I speak from experience. When we started with dengue NS1, NS1 was not in any part of medical literature back in 2008 and 9. There was very
right.
little knowledge around that. So it took a radical idea, and eventually it helped in improving patient outcomes, right?
Yeah. Any
So
day, any day. So
thank
when I talk about, I say that look for what exactly we are getting up. We are definitely asking for a reasonably good amount of why paired blood cultures it started. Then paired people started only aerobic cultures. Why not anaerobic? Why to miss it? Then we talk about MIC values. We are not discouraging to have distribution. Definitely it should be there. There are conventional methods, but at the same point of time, MICs can be very helpful for the clinicians to taper the doses, look for the diagnostic, for the therapeutic drug monitoring.
All right. On that note, I think it's a nice note to close on. That look, don't disregard the old, but you need to look forward, and you need to look at what
I belong to old school of thoughts. Definitely, old will never win over the newer ones. Though we have technologies, but old is gold.
AI is there, and AI has a habit. I learned it to my utter dissatisfaction today about hallucination. So AI also hallucinates.
If everything if everything falls apart, so much of diagnostic automation things are there. If everything falls apart, so we still have the conventional hand methods. We can still follow that. There, none of us will fall through.
Absolutely, ma'am. Well, thank you so much for your
Right.
attention to detail, ma'am. And we look
Thank
forward
you
to
so much.
meeting you again very soon, and we'll talk about antimicrobial resistance in that session, right? Thank you.
Hopefully, right, right.
Forward
Thank
to
you
it.
so much.
Thank
Thank
you.
you, everyone. Thank you so much.
Thank you all for joining us today. We'll see you soon next week on the 7th with a discussion around scrub typhus. Till then, Jai Hind.